continuous improvement collaboration. Today we have a special guest with us who will be lead this session and sharing their valuable
of Chemistry in Vishnu Institute of Pharmaceutical Education and Research. And today I'm going to deal with the topic computer-aided drug design. Coming to the topic, the overview drug discovery process. In this, we are discussing about the overview of computer-aided drug design, computer-aided drug design approaches, what is molecular docking, approaches of molecular docking in drug design, success stories in molecular docking, and conclusion. So coming to drug action, first you have to know what is a drug. So it's any substance used in the diagnosis, treatment, or prevention of a disease or a compound of a medication. So here we can see how the ligand is binding to the protein site. So when this ligand binding to the protein site, we are getting the protein with binding ligand. So then only it can show the action. So here the drug, it may be natural, synthetic, or semi-synthetic. So how these drugs are developed generally? It's just a cartoon to find out a particular drug. So this scientist is telling Bangladesh, it took all over 65298 seconds to discover a particular drug. So you can calculate how much time it is taking to introduce a new drug into the market. So coming to the drug discovery approaches, we are having the serendipity, which is pure luck, random screening, chemical modification, rational drug design. So coming to the serendipity, so here the workers in TNT factories always complain from headache due to dilation of the blood-brain vessels. The trinitrotoluene was the basis to prepare nitro derivatives, which were used in angina to dilate coronary blood vessels and elevate pain. So by this, how these TNT factories, how they are suffering with dilation and blood-brain vessels. From that, the scientists introduced the nitro derivatives, which used to treat angina to dilate the coronary blood vessels. So that is one serendipity approach for the drug discovery. And next one is the mustard gas tanks used in the Second World War exploded in Italian harbor. So they discovered that the person who suffered and inhaled this gas lost their defense against microorganisms due to the destruction of white blood cells. So the person who was affected with this mustard gas, the white blood cells are decreased and frequently they are undergoing infections. So from this mustard gas can be used in leukemia to inhibit excessive proliferation of white blood cells. Like that only warfarin also used as a rat poison. And we all know that the famous antibiotic penicillin, it also got discovered due to this serendipity model only. And coming to the random screening a large number of synthetic chemical compounds or natural products for desired effect. So it is successful in past, but not ideal for a number of reasons nowadays. It is inherently repetitious and time consuming. It is trial and error approach. One does not need to know the structure or mechanism action of the drug, nor the structure of the target upon which the drug will act. So in random screening, randomly we have to select different drug candidates and we have to go for its activity. So in such cases, no need to know the structure of the drug or its mechanism of action, or no need to know the structure of the target protein also in random screening. So coming to the chemical modifications, in chemical modifications, we have to know the lead generation. We have to identify the lead and we have to go for the lead generation. It may be natural or upon the screening of the data. Next, going for the biological testing of the lead molecule. After biological testing, we can go for the drug design cycle in which the lead modification can be done with the synthesis of the new compound. Like we can go for the SAR study, structural activity relations of the lead molecule. In the biological testing, if the lead molecule gets promising results, we can go for the preclinical studies. So here the long road to the new medicine. So to discover a new medicine, here first we have to identify a new disease. A new disease came into the world. A group of scientists, they have to discover, and after they have to discuss, and after the discussion, they have to discover which protein is responsible for that particular disease. So to identify that particular protein and to isolate the protein involved in the disease, it is taking almost 2 to 5 years. So after isolation of the protein, again going for the find the compound effective against the particular protein. There's a hit ID to lead. Again, it is taking to identify the particular drug candidate. It is again taking 2 to 5 years. After identifying the lead molecule, again you have to go for the preclinical testing, animal efficacy lead optimization. It is again taking 1 to 3 years. So after crossing all these barriers, going for the formulation development. After formulation development, you have to go for the studies in the healthy volunteers, safety, and POM. So in POM, phase 1, phase 2, and phase 3, again covering all these phases, it is taking around 2 to 10 years. So after passing all these barriers, candidate medicine tested in 3 to 10,000 patients in phase 3. So after crossing this phase 3 level, again, we go for the application of the NDA, FDA approval, which is taking almost 2 to 3 years. So to discover a particular drug, it is taking almost all 10 to 15 years, the lifetime. And again, it is more costly. It is almost 500 to 800 million US dollars. So to overcome all these, we are having the computer-aided drug design. So this is also one more model. You have to test a lot of compounds to find a particular drug. So in the initial stages, you have to test at least 1 million of, from that you are getting only the last stage in the phase 3, you are getting only one drug. So it is taking 10 to 15 years, and it is highly costly, 500 to 1,000 million US dollars. You have to spend more money to introduce only one drug by the older methods. So that is why we have to use this computer-aided drug design. So why we have to use the computer-aided drug design? Because the future is bright, the future is virtual. That is why you have to learn the computer-aided drug design. So all nowadays, the world is going into the artificial intelligence also. So that's why we have to learn this computer-aided drug design. So in this, we have to learn about, we have to get the knowledge about the virtual screening, where hit identification is identified, and they select the lead compounds. After selecting the lead compounds, we have to optimize the lead compound. So the major advantages of the learning with CADD is it reduces cost and time, reduces animal use in the experiment, improves success rate, improve understanding of drug-protein interactions. And the major advantage is it's saving our 2 to 5 years of time and 10 to 500 million US dollars in the introduction of the new molecule or the new drug. So these are the more benefits why we have to learn the computer-aided drug design. Majority it is saving our time and money also. So methodologies and strategies of CADD. So in computer-aided drug design, we are having structure-based drug design, in which we have to know the protein ligand docking, protein modeling, molecular simulation. So next one is ligand-based drug design, and this structure-based drug design is a direct method. First, we have to identify the protein. We have to crystallize the protein. We have to go for the 3D structure of the protein. We have to know the 3D structure. And again, we are going for the protein ligand docking. That is a direct method. And ligand-based method, which is indirect method. We don't know the structure of the protein. We don't know the structure of ligand. Just on the database, which is available from the database, we'll go for the ligand-based drug design, which is similar analysis, QSR model, pharmacophore models. Next, ADMET prediction. So by using this CADD, we can go for the ADMET prediction also, adsorption, distribution, metabolism, elimination, and toxicity. We are having the different softwares available in the online. Go for the computer-aided drug design. So coming to the structure-based drug design. So discovery or design of molecule that interact with biochemical target of known 3D structure. So here our biochemical target, it may be enzyme, it may be protein, or it may be any DNA, maybe monoclonal antibodies, whatever it may be, which is a biochemical target. So in this, we are targeting determine the protein structure. Know the structure base, we have to determine the protein structure. We have to identify the interaction sites of the protein. From that, active sites, we have to design de novo synthesis design and 3D base. From that, we have to evaluate the structure, what we have interpreted. After evaluation, you have to go for the synthesis of the drug candidate, which is suitable to that active site, to bind to that active site. After going for the test candidate, from that, we have to develop the lead compound. So these are how we are doing that structure-based drug design. In structure-based design, all is the first step, where we have to go for the experiment or the sequencing of the protein molecule or the receptor molecule. We have to identify its sequence models. After that, we have to go for the crystallization. After crystallization, we have to derive the protein 3D structure. So when you got the protein 3D structure, by using different software, we can analyze its binding pocket, the active site of the 3D structure of the desired protein or the targeted protein. Next is structure-based drug design. So in this docking, so first the best match between the target protein and drug candidates. So this is the drug candidate and this is the targeted protein. So how best this ligand is going at binding to the active site of the targeted protein. Here we can see. So here in the animation, we can see the best matching. How this our ligand is going at binding to the target protein, the active site of the target protein. So that is what we are doing in the docking studies. So for these docking studies, we can use the softwares Autodock, Glide, Cybele, MOE. These are the available softwares. By using these softwares, we can easily do the docking studies. The ligand-based drug design is an indirect model. So where the molecule shape is matching, millions of molecules per day are introduced by using this ligand-based drug design. So by using the pharmacophore model studies, we can design the ligand, where if we don't know the structure of the protein, or if we don't know the 3D structure, we don't know the active sites, we can go for indirect method, ligand-based drug design. So coming to the molecular modeling, the term molecular modeling expand over the last decade from a tool to visualize three-dimensional structure and to stimulate, predict, analyze the properties and the behavior of the molecule on an atomic level to data mining. So from we are studying by using this topic molecular modeling from the atomic level of the drug candidate to the data mining. So it is a platform to organize many compounds and their properties into database and to perform virtual drug screening via 3D database screening for novel drug compounds. So molecular mechanics. In molecular mechanics, it refers to the use of classical mechanism to model the geometry and motion of the molecules. So how the molecules, how the ligand is going and docking at the active site of the protein, how its geometry is changing, how the motion of the molecule is changing. It is just like induced fit theory. For the best fit of the ligand into the receptor molecule, how the 3D conformation of the ligand is changing. After changing the 3D conformation, how which geometry is best fit into the receptor molecule. So in molecular mechanisms, we got one. And in molecular mechanism methods are based on the following principle, where we all know that the nuclei and electrons are now put into atom-like particles. So these atom-like particles are spherical and have a net charge. So interactions are based on the springs and classical potential, where when the atom is having a particular net charge, it can easily undergo the interactions which are based on the springs and classical potential of that atom. So interaction must be pre-assigned to specific set of the atoms. So the specific set of the atoms which are present in the receptor site and the ligand site, we have to assign a particular interactions. So these interactions determine the spatial distribution of atom-like particles and their energies. By using the molecular mechanism, we can determine all this. We can go into the atomic level of the ligand and proteins, and we can predict how they are interacting, how the interaction is taking place. And in structure-based library screening, so what do we need? So we need mainly the compound libraries, protein targets, binding site in the protein, docking, generate different possible conformation of the compounds in the binding site, score, evaluate the strength of the protein ligand interactions. There's a docking score, and select preferred ligand to propose a list of prioritized compounds for experimental screening. So in the nowadays, in older days, the drug targets, mainly the drug targets, here you can see the molecular the structure within the organism linked to a particular disease, whose activity can be modified a particular drug. So that is our drug targets. It's a distribution of the drug target by biochemical criteria. Here, the major portion is occupied by the enzymes. The scientists are targeting major part with the enzymes, and next is occupied by receptor, which is very less comparatively. The target of the enzyme is more when compared with the receptor, which is only 4.60%. And followed by monoclonal antibodies, factor, regulator, and hormones, binding proteins, transport proteins, nuclear receptors, structural proteins, signaling proteins, ion channel, RNA, and lipid binding proteins. So these are all comes under the targets where a particular ligand can be designed and we can inhibit a particular disease. And in biochemical criteria, these are the biochemical targets. And when coming to the therapeutic area, here we can see in therapeutic area, the major target is neoplastic diseases. Nowadays, we all know that the cancer is the leading disease in the world. So the scientists are majorly focusing on the neoplastic disease, hormones and hormonal antagonists, viral infections, blood-forming organisms, immunomodulations, bacterial infections, renal and cardiovascular functions, synaptic and neural effectors, junction sites and central nervous system, parasitic infections, and followed by the different target sites. This comes into the distribution of the targets in therapeutic area. That is in compact. When the distribution of the targets is biochemical, we can see the major portion is enzymes here, but here the major portion is the neoplastic diseases. But in nowadays, the drug targets in the current therapies, you can see here, the major portion is occupied by the receptor, which is 45%. But in older days, the major portion is occupied by the enzymes, which is 77%, but now it is only 28%. So by using this computer-aided drug design, the major site of our target is receptors. So here, the drug targets, when you are targeting a particular enzymes, we have to identify its inhibitor. When you are targeting a receptor, we have to design a synthesis agonist or antagonist. When you are targeting an ion channel, we have to introduce the blockers. You are targeting the transporters, inhibitors, DNA, which you are targeting, we have to synthesize blocker. So this is the information is used by the bioinformaticians to narrow the search in the group. So by using all this data, the bioinformaticians, they are just narrowing the search in the group. They're not going for the broad search. They're just narrow search with the help of all this data. So these are the examples of the enzymes, what you are targeting and what is the activity of the drug and the examples of the drug. So here we can clearly see What are the mainly we are targeting the oxidorejectases, aldehyde in the again we are having the aldehyde dehydrogenase, MOAs, cyclooxygenases, and vitamin K epoxidase, aromatase, lipoxidase, different types of enzymes we are targeting. And here these drugs, how they are acting on these particular enzymes. It is clearly mentioned in this table. So now coming to what is CADD. So first we have to do what is computer-aided drug design. So it is a computational chemistry, is the chemistry whose major goal are to create efficient mathematical approximations and computer programs that calculate the properties of the future drug molecule or the drug molecule which we are going to synthesize. There is a future drug molecule and thus helps in the process of the drug design and discovery. So it is simply the mathematical approximations or the mathematical approaches in combination with the computer programs. So the drug discovery today are facing a serious challenges because of the increased cost and enormous amount of time taking to discover a new drug, and also because of various competition on the target, target identification and target validation. So next is lead discovery. So what is the lead molecule? We have to discover that one. After discovering the lead molecule, we have to go for the lead identification, and we can done the lead identification by database filtering because we are having a lot of data available in the books or in the internet. From that, we have to filter the database and we have to choose which pharmacophore is suitable for that particular target. And we have to align that particular lead molecule and similarity analysis we have to do. And we have to do the bioforce, very high output screening we have to do, and diversity selections. So after doing all this, we can go for the lead identification. So after lead identification is done, again, we have to go for the next step, lead optimization. So in optimization, we go for the QSR, ADMET, combinatorial libraries, de novo design. So by doing or by using this computer-aided drug design, again, we are mentioning here, it's saving 12 to 15 years of our time and cost is 500 to 18 million US dollars of money. So molecular docking. Here, docking is the identification of the low energy binding modes of the small molecule or ligand within the active site of a macromolecule or receptor whose structure is known. So here, structure-based drug design we are doing here. If we know the 3D structure of the macromolecule or the receptor, so by using this molecular docking, we can identify the low energy binding modes of the small molecular ligand within the active site of that macromolecule. So docking is the computational determination of binding affinity between molecules, maybe protein structure and ligand that may be protein or DNA, like the different models we are having in docking. So given a protein and a ligand, find out the binding free energy of the complex formed by the docking them. So why we have to go for the modeling? So by doing this modeling, the experimental determination of the structure is still a time-consuming, expensive process. So number of known sequences are more than the number of known structures. Structure information is essential in understanding its function. So that's the reason why we have to go for the modeling. So by doing the molecular docking, the classification is the docking or computer-aided. It can be broadly classified into the first one is receptor-based method, making use of the structure of the target proteins. So here you can see the receptor-based method. We know the receptor, we know the binding site, active site of the receptor, and here the ligand is blindly going in, binding at the receptor site. So that's the receptor-based method, making use of the structure of the target protein. If you know the structure of the target protein, you can go for the receptor-based method. So if you don't know the target receptor, but we know the structure of the ligand, so ligand-based method. So we have to select a particular ligand from the different database mining. So from that, again, we have to go for the hit and model. So ligand-based model, we have to go for the based on the known inhibitor. You know the inhibitor. Inhibitor is our ligand only. So if you know that inhibitor, from that inhibitor, again, we have to go for the lead optimization and structure activity studies, and we have to go for the hit and run model. Next, receptor-based models. So use the 3D structure of the target receptor to search for the potential candidate compound that can modulate the target function. These involve molecular docking of each compound in the chemical database into the binding site of the target and predicting the electrostatic fit between them. The compound are ranked using an appropriate score, a docking score function such that the scores correlate with the binding affinity. Receptor-based methods has been successfully applied in many targets. And here the ligand-based strategy, in the absence of the structural information of the target, we don't know the structure of the target protein or enzyme. The ligand-based method make use of information provided by the known inhibitor from the target receptor. So structures similar to the known inhibitors are identified from chemical databases by variety of methods. So we are having a large chemical databases. So from that, by using different variety of methods, we can identify the structure similarity of the known inhibitors. So some of the methods widely used are similarity and substructure searching, pharmacophore matching, or 3D shape matching. So numerous successful applications of the ligand-based methods have been reported. So by using these ligand-based strategies, so without knowing the structure of our target, by just knowing the similarity structure or substructures, pharmacophore matching, or 3D shapes, we can successfully application of ligand-based methods have been reported. So we can design a new molecule by all these strategies. So basic binding mechanism. In this basic binding mechanism, the complementarities between the ligand and the binding site. So the steric complementarity, that is the shape of the ligand is mirrored in the shape of the binding site. And we have to identify the physical chemical complementarities also. In the categories of the docking, we are having the protein-protein docking. Both molecules are rigid. Interaction produces no change in the conformation because the proteins are very rigid. So we cannot have this change in the conformation in protein-protein docking. Protein ligand docking. In protein ligand docking, ligand is flexible, or the drug candidate is flexible, but the receptor protein is rigid. So always remember the receptor protein is always rigid, so it cannot change its conformation. Only the ligand molecule in protein ligand docking only it is flexible. So the ligand can change its conformation. So the interaction produces conformation change in ligand. In protein-protein docking, you can see here the both are rigid. The computational model of the quaternary structure of the complex is formed by the two or more interacting biological molecules. So here A and B are the two protein molecules. So they are interacting with each other. And they're forming a biological macromolecule, AB. The protein-protein complexes are the most commonly attempted target of such modeling, followed by protein nucleic acid complexes. Coming to the protein ligand docking. In protein ligand docking is to predict the position and orientation of the ligand when it is bound to a protein receptor or enzyme. So here this is our protein and this is our the small molecule ligand. So this ligand is going and binding at the active site of the binding site of the huge complex, a huge protein, and forming the complex. So here clearly we can see the binding site where our small molecule, small ligand is going and binding here. So this is protein ligand docking, and here the ligand is not rigid. It can change its orientation. So what are docking and the scores? To place a ligand into the binding site of the receptor in the manners appropriate for optimal interaction with the receptor. To evaluate the ligand receptor interaction in a way that may discriminate the experimental observed mode from other and estimate the binding affinity. So that is docking, and it is a binding affinity score. So first we have to allow the ligand and the receptor for docking, and we are getting the docking complex. After docking complex, how much amount of energy is required for the docking. So that is what the docking score. So this docking score we can predict by using the Autodock software, or different types of paid and non-paid softwares are available. So again, we can predict the structure of this complex by using the X-ray analysis methods. And here available docking programs, Schrodinger, Accelerator Pro, Gold, Dock, MOE Dock, Flex, Autodock, Freed, Hammerhead, Argus Lab. Those these are already in the first day of the FDP. Already Sarah mentioned all this. And the components of the docking software. So here, what are the components of the docking software? How this docking software is working? So typically, protein ligand docking software consists of two main components which work together. The first one is the search algorithm. So generate a large number of poses of the molecule in the binding site. So in search algorithm, it will search the large number of poses, in which pose the ligand is going and binding to the binding site with least energy. So it will have to generate a large number of poses, different poses. It has to go and bind at the binding site. So from the binding site, when it is going and posing binding, we'll get the score functions. Calculate the score or binding affinity for particular pose. So every pose, it is calculated the scoring functions. So at which pose the scoring the docking score is less, that is the best pose for docking. The binding and affinity of the score represent the strength of the binding. So it is the binding affinity or the score represent the strength of the binding. So it's coming to the docking flowchart using Autodock. So how we have to perform the Autodock. So this is best docking flowchart. The first database of the ligand protein. We have to get the structure of the ligand, and we have to get the structure of the protein from the databases, which is available online, or we can make it by using different software like Camera, Mole, or CamSketch, like that. So after getting the structures of the ligand and protein, so the protein is converted into protein PDB. We have to save the protein in the form of PDB format, protein data bank format. After getting the protein PDB format, again, we have to give some charge. Some amount of the charge is to give into the protein. Again, we have to save that protein in the PDB QT. QT is the charge format. So we are using some charge, we are giving some charge to the protein. So the charge required here is to interact with the ligand. So that is why we have to give some charge to the protein. So again, ligand also we have to save in the PDB format, protein data bank format. Again, to the ligand also we have to give some charge, maybe Coleman charges. So after getting the protein dot PDBQT format and ligand PDBQT format, then we have to go for grid generation. So we'll get the protein ligand GPF. After grid generation, grid parameter file we are generating. From grid parameter file, we'll get the map files. So this is auto grid ligand protein GLG, grid log generator file. After allowing it to go for the docking, we'll get these formats, GPF format, GLG formats, and ligand protein DPF format, docking parameter file. So there are the different stages in docking. So after getting that docking parameter file, now it is allowed for final docking. After going for the auto dock, we'll get the protein ligand protein DLG format, dock confirmation, dock log generator. So after getting the dock confirmation ligand protein DLG format, now I'll get the different scores. In which pose the docking scores it is generated, and in which pose we are having the least docking score, that is the good affinity side or the good binding energy side. So here the first step, the ligand preparation for the docking using autodock. In the ligand preparation, first you have to assign the charges. After that, assigning the charges, we have to go for define rotatable bonds. These rotatable bonds are nothing but how many single bonds are available in the ligand, the design ligand. Next, rename aromatic carbons. So how many aromatic carbons are there? We have to rename that one. After all this, we have to generate the PDBQT format of the ligand file. So the first step in the preparation of the ligand. Again, preparation of protein using autodock. So after downloading the 3D structure of the protein from protein data bank, so we have to add essential hydrogens. After adding the essential hydrogens, we have to load charges. Maybe Coleman charges generally used, or Gassier charges. Depending upon the requirement, different charges are available. We can load the charges. And we have to remove the water molecule because we all know the water molecule is polar. So when this water molecule is present, it may interact with the binding or it may interfere with the binding affinities. Next, after removal of the water molecule, and here in the protein 3D structure, we have to repair that protein 3D structure also, because any missing proteins are there. By using autodock software, we can repair that missing amino acids also. So after doing all these steps, again, we are to generating the PDBQT protein file format. So here the ligand PDBQT format is generated and protein PDBQT file also generated. The next step. So here you can see, this is how the protein is visible when you are using the autodock. The visibility of it is the autodock tool. So when you are giving the PDB format into the autodock tool, this is how the protein is visible. And this is how our ligand is visible. So coming to the applications, so determine the lowest free energy structures for the receptor ligand complex. So our main target is how much low energy the ligand and receptor is taking for forming the receptor ligand complex. The energy is less, that is the best binding pose. And you search database and rank hits for lead generation. And calculate the differential binding of a ligand to different macromolecular receptors. Study the geometry of a particular complex. Propose modification of the lead molecules to optimize potential or other properties de novo design for lead generation. And we can go for the library design also. Screening for the side effects that can be caused by the interaction with other protein like proteases, cytochrome, P450 and other can be done. So by using this, we can predict the toxicity studies. It's any like cancer-induced metabolism also, we can screen the side effects by using the autodock studies or targeted drug design. It's also possible to check the specificity of the potential drug against homologous proteins through docking. So by doing the homologous protein docking studies, it is possible to check the specificity of the potential drugs. So docking is also a widely used tool in predicting protein-protein interactions. So knowledge of the molecular association aid in understanding a variety of pathways taking place in the leaving and in revealing of possible pharmacological targets. And docking-based virtual high throughput screening. So virtually we can go for high throughput screening. And moreover, the major advantage is less expensive than the high throughput screening, faster than conventional screening, scanning a large number of potential drugs like molecules in very, very less time. High throughput screening itself is a trial and error approach, but cannot be better complemented by virtual screening. Just we are going many times, we can go for the trial and error approaches by using this computer-aided drug design. So growing evidence of the success. So by using this computer-aided drug design, so what are the success rate of this computer-aided drug design? So here, these are the drugs and the target and the disease, how successful this computer-aided drug design is. Dorazamide is a carbonic anhydrase inhibitor, and it is used in the diuretic to treat the diuretic. Sisconavir, HIV protease, it's used to treat in the treatment of AIDS. Relenza, urothymidase, it is used in the treatment of AIDS. We are having AG85, AG337, AG331, thymidylate synthetase used in the cancer treatment. So these are the growing evidences of the success in computer-aided drug design. So discovery of Idinavir, the HIV protease inhibitors, identification of haloperidol as a lead compound in the structure-based design for non-peptide inhibitor in HIV, carbonic anhydrase treatment of glaucoma, renin treatment of hypertension, dihydrofolate reductase, antibacterial, neuraminidase, antiviral, HIV aspartic proteinases, acquired immunodeficiency syndrome. So these are the growing evidences in the success rate of by using computer-aided drug design. So coming to the conclusion, molecular docking used the promising effect of identification and optimization in modern drug discovery. The combination of the chemical information of natural product with docking-based virtual screening will play an important role in drug discovery in the post-genomic era as more and more new potential targets are emerging from the functional genomic studies. So docking-based virtual screening lead to much higher hit rate than traditional screening methods. So the conclusion, docking method provides an opportunity for the designing of active compounds. Because by using computer-aided drug design, there are a lot of opportunities there to design the new active compounds. However, it hasn't emphasized that docking-based virtual screening is not the replacement of the actual experimental screening. As a matter of fact, these two methods are highly complementary. And coming to the future directions of the CADD, the pharmaceutical history indicated that natural products provide a large number of drugs to the market. But even for the currently used drug target available, natural products have not been tested completely. So in the nature, a lot of compounds are available which has not been tested till date. So by using this computational medicinal method can contribute its unique role in achieving the task of examining the interactions of all existing natural product with all possible targets. So by using this computer-aided design, we can analyze the natural products also with all the possible targets. So this is the future direction of the computer-aided drug design. So any questionnaire regarding this topic?
Thank you, sir. Thank you for your valuable information. Any questions?
My dear participants, if any questions, I'm ready to answer here. Anybody having the queries, you can contact me personally also.
So can you explain once again docking procedures? Sir?
Docking procedure, madam?
Yes.
Yeah, sure. So in docking procedure, the first step is we have to the ligand preparation. So ligand preparation for docking using Autodock. In ligand preparation, first we have to draw the structure of the ligand by using ChemDraw, or we have to download it from the online sources also. So after getting the structure of the ligand, we have to go for assigning the charges to that particular ligand. We have to convert the ligand into the PDB format. After converting the ligand into the PDB format, we have to assign the charges. So because the charges are responsible for the affinity. If the charges is present in the ligand, then only it can go and bind with the active site of the receptor. So that is the reason why we have to assign the charges to the ligand. So after assigning the charges, we have to define the rotatable bonds. So how many rotatable bonds are there? Because these rotatable bonds are responsible for the flexible nature of the ligand. So we have to define how many rotatable bonds are there in the ligand what you have chosen. So that is why define rotatable bonds. Here the rotatable bonds are nothing but generally the single bonds. The single bonds will be responsible for the orientation or the 3D geometrical structure of the ligand. So if more number of rotatable bonds are present, it is more flexible. The ligand is more flexible. And rename the aromatic carbons. So generally we are having aromatic groups are present in the ligand. In that, how many aromatic carbons are there? We have to rename that one. And after renaming the aromatic carbons, we have to go for the generate the .pdb qt format. So already I told that after assigning qt is nothing but the charge. After giving the charge, we have to generate the pdb qt ligand file. After that, preparation of protein using Autodock. Add essential hydrogens. Similarly, how we prepare the ligand. First, we have to select what is our target protein. So after selecting the target protein for the particular disease, we have to download the protein 3D structure from the PDB protein data bank, which is available online. RCBS, we can directly download the 3D structure of our required protein, target protein. So after getting the 3D structure, we have to optimize the 3D structure because the 3D structure, when they go for the crystallization, different pro-crystals are present, water molecules are present, ligands are present. So all these, we have to remove all these unwanted data from the protein 3D structure. Because if all these 3Ds, all these are present, they may interfere with the ligand and protein binding. So that is the reason why we have to optimize the protein also. In optimization, we have to remove the water molecules. Here I mentioned, remove the water molecule. We have to load the charges. We have to add essential hydrogens. Generally, we have to add polar hydrogens. After doing all that, and we have to remove the heteroatoms from the protein. So after doing all this optimization, the final step is we have to generate the PDB-QT of the protein file. So after the first step, we are generating the PDB-QT of the ligand file. In second step, we are generating the PDB-QT of the protein file. So after generating these files, when you allow it into the Autodock software, like this, they will appear. So this is the macromolecule, which is the protein or is the targeted protein. And here you can see a green color small benzene-like structure. Okay, is it visible? The benzene-like structures in green color. Rest all are in blue and white color. The blue and white color, that is the structure of protein, the targeted protein. But here the green colored, which is embedded inside the protein, that is the structure of our ligand. So this is, if you eliminate the structure of, if you mark the structure of the protein, we can see the structure of the ligand. These are the structure of the ligands. And these straight chain-like structures, these are the structure of the amino acids. These are the binding sites of the amino acids. So these aromatic ring structures, they are our ligands, and how these ligands are binding with different amino acids in the binding site. That clearly it is visible. So it is DPN 12 active site of TY450 and DPN 11 active site of glycine at 186. The position of the glycine which is present at 186. So these are the applications. Here also you can see. So after getting ligand PDB-QT and protein PDB-QT, we have to generate the grid formatting, that is ligand protein GPF, grid parameter file we have to generate. After generating the grid parameter file, so we have to allow it for the auto grid ligand protein GLG, grid log generator file. There are the different files by using the Autodock we can generate. By giving different commands to the Autodock software, we can generate all this easily. So after getting the grid log generator file, and here also we are getting the ligand protein DPF, docking parameter file. So all these again allowed for the complete auto docking process. So after allowing this auto grid GLG and ligand protein DPF format, so finally you get ligand protein DLG format. This is the docking confirmation we will get. So after getting the docking confirmation DLG dock log generator, so we'll get the docking score also. So from the docking score, almost we'll get the 10 docking score. From the 10 docking score, we have to choose which one is the least one. That least docking score reflects that is the best energy taken by a best pose taken by the ligand or the receptor for its forming of the binding site or the complex. Is it clear, madam?
Clear, sir.
Actually,
Any
just by seeing this, you may not be understood. But if you are practicing that by using the software, it is very easy. But for that, we need a workshop for that. If you are attending workshops regarding this Autodock software, and nowadays Autodock next level has came, that is molecular modeling. Nowadays, recently you have the molecular modeling and molecular simulations also came. Nowadays, many of them are using these molecular simulations. Instead of going for the molecular modeling, the next level is molecular simulations. So in the simulations, we can clearly see, we can see the video of the simulations, how this ligand is going and binding to the receptor side.
Hello. Hello.
Yeah, hello, sir.
Tell me, sir, what is the current situation in our your topic? What is the current situation in our pharmacy market? How to use that?
So already current situation, I mentioned of the success rate.
Yeah, success rate. What is the percentage?
The percentage is very least only, sir. But
Sir?
nowadays it's very less only the percentages. But
Yeah.
still, it is in developing stage only. In recent almost a decade, from the decade, we are using this computer-aided drug design. But we need more and more data has to be collected and more software has to be developed. Recently, nowadays, involved in it, means in recent days, the scientists are using molecular dynamic simulations. Now, this is the latest version.
Okay,
So
sir.
that-
In academic
this-
side, how to use? In our recent, you can tell the industrial point of view in our. I'm asking, what is here this? Academic side, how to use?
when we are doing our PhD or research work,
Yeah.
before going to the synthesis of the particular structures,
Okay.
first we have to just design the structure. After designing the particular structure, you go for the in silico studies by using this CADD softwares. So from the software, we can, so for suppose if you are targeting a particular disease, for a particular disease you are targeting, we have to identify what is the protein or what is the receptor responsible for causing that particular disease.
Okay.
So after identifying the protein, again now we are designing a particular ligand,
Okay.
right? You are having both the structure of the ligand and you have identified the protein which is
Okay.
responsible.
Okay, I can understand. Tell me total
Yes.
conclusion part. Why I have to study? So why simply say in two or three lines? Just will tell me what are the total conclusion part. Why I have to study? How to use that in current situation for student and research scholar?
Yes, sir. Already it's clearly mentioned by using CADD,
Okay.
we can save a lot of time to discover a particular drug.
Okay.
So, and we can save a lot of money,
Okay.
both time and money as we are saving by using this computer-aided drug design. If you are getting the best results in computer-aided drug design, we can go for the synthesis of the particular drug for which we are getting the best results in CADD. If you are not getting
Okay.
CADD,
But
we
sir,
can
sir,
predict that one.
please listen. In our colleges, there is no facilities. Okay, just we'll study how to improve that. So we'll go through the commercial point of view or attachment to the industrial point of view like this.
So actually, what I can suggest you is first the basics. Many free softwares are available, like you can go for Autodoc 4.2 or Swiss ADME studies, OCD properties studies. They're free of cost, which the academician can easily access that one. So if you have any fundings or if you have any research fundings for your work, then you have to go for this paid software like Schrodinger, Glide, Gold software like that.
Okay. Thank you, sir. Nice.
So for academician, if you're having that much funding support, you can go for the paid software, sir.
Okay. Thank you, sir. Thank you. Good. Super.
Yeah, okay, thank you very much, sir.
Thank you.
Hello.
Yeah, hello.
Good morning, sir.
Yeah, good morning, madam.
Sir, my question is, can we relate the docking score of a derivative with the results of biological activity? How
Yes,
can we
yes,
justify if the
but-
results does not match?
yes, but in many cases, that is also happening. We cannot completely believe in the score what we got in the computer-aided drug design. We have to repeatedly go for computer-aided drug design, whether you are getting the particular score is correct or not. We have to validate that one. After validation only, we have to go for the synthesis and we have to go the in vitro activities. So first we have to validate the score. For validation of the docking score, again, we have to use for the molecular dynamic simulation, whether your design ligand is going and targeting your specific target site or not. So after validating only, you have to go for that one. And after that, you can get the successful result.
Okay.
So how can we do after synthesis, sir? Docking
Sir?
studies. Can we do docking after synthesis?
After synthesis also we can do, sir. Just for docking, we just need the structure of the synthesized drug and the structure of the targeted protein. That is enough. Before synthesis also you can do, or after synthesis also do. But
Yes,
after
sir.
synthesis, if you do means, of course, you already synthesize the particular series of the drugs, they may give the activity or not. But before synthesizing that particular drugs, if you do, then you can predict whether synthesized drugs are active or not by using this computer-aided drug design. Just we can have the prediction whether the design drug is showing the good activity or not.
Yes, sir. Yes. Thank you, sir.
Thank you very much, sir.
Sir, in docking process, how can we convert into 2D and 3D forms, sir? 2D and 3D structures.
2D, and after completing the docking studies, by using different softwares are there, madam, like PyMol is there to analyze the docking Autodoc score. We are having other software like Discovery Studios, BioVia. By using all this, just we have to generate the complex of the Autodoc. By taking this complex, by allowing the complex into these different softwares, we can get the 2D and 3D structures of that Autodoc score.
Thank you, sir.
So
Very
once you practice, this Autodoc is very easy.
Sir?
Once if you practice regularly, Autodoc is very easy. Even students can easily learn this one. Compared to the scoring, the paid softwares and monitor dynamic simulations, Autodoc is very easy.
Thank you, sir. Any questions?
So, and one more question, what is the difference between Autodoc and molecular simulations? So, simply I can tell you Autodoc is just a basic software where you're getting only the docking scores. But in molecular simulations, directly we can visualize how this ligand is going and fitting at the particular target site or binding site. In simulation, the word itself is how the ligand is simulating, how the protein is simulating with the ligand, designed ligand. So clearly we can see there is an advanced next level of the computer-aided drug design, molecular simulations.
What are the free softwares for simulation, sir?
Actually, for simulations, we are having software is there. Gromax software, free software version is available, sir.
Okay, sir.
Gromax. But to run the molecular simulations, the computer software events, the data, what you can say is the RAM and the hardware motherboard, it should be in high end only. The normal laptops or the basic laptops, they cannot properly run these molecular simulations. It needs a high end computer system.
Okay, sir.
If you're having the high end laptops or the high end systems, then easily you can run the molecular simulations. Or if you're using a normal laptop, easily it can get hang out.
Windows or Linux, sir? This one.
Yes, Linux, sir.
Linux.
Just you have to install the Linux to run the molecular simulations. After installing the Linux, again you have to install the Gromax simulation software. That we have to go for the simulations. But for that we need a high-end laptop only.
Okay, sir.
Any questions? Thank you, sir. Thank you for your valuable information. Docking
Actually,
studies.
I have to say thank for your principals or organizing members for providing such a wonderful opportunity as a speaker in your esteemed college for this FTP. Really, I'm very thankful to you for providing me such an opportunity. And I'm always ready to organize such events here also.
Definitely, sir. Definitely. Thank you so much, sir. Very impressive. Docking
very
and
much.
molecular simulation studies. Thank you so much, sir. Now, I would like to invite Pawan Kumar, sir, Assistant Professor, Department of Pharmaceutical Analysis. He will proceed with vote of thanks.
Good morning, sir. Myself, Pawan Kumar, Assistant Professor, in the Department of Pharmaceutical Analysis and Chemistry. So, it is my privilege to deliver the vote of thanks on behalf of his College of Pharmacy. I would like to extend our heartfelt gratitude to our extended speaker, Dr. V. Muralidharan Garu, for their insightful and thought-provoking section. Your knowledge and experience have added immense value to this program, and we are deeply appreciative of the time and support you have dedicated to being here with us. So I would like special thanks to our principal, sir, Dr. M. Kishore Babu Garu, for his continuous support and encouragement in our organizing this program. Your leadership and vision have been instrumental in making this event as a success. I would also like to thank to IPGA President Garu, Dr. V. Vijay Bhaskar Garu, Annapareddy Garu, General Secretary, Dr. P. Srinivas Babu Garu, National Joint Secretary, Dr. Sri Lakshmi Garu. A special thanks goes to our coordinator, Dr. Swati Garu, our organization and our organization committee. I would also like to acknowledgement and that all our faculty members and participants for their active participation throughout this section. Your willingness to learn have made this program very much dynamic and interactive. Finally, I would like to express our appreciation to the technical team who ensured that the online platform. Once again, thank you for all presence and participation. We hope that the knowledge, insights gained during this FDP will be beneficial in your teaching and research endeavor. Thank you. Thank you so much, sir.
Thank you very much, sir.
Thank you, sir.
Thank you, sir. Thank you
Thank
so
you,
much,
ma'am.
Leader, sir.
Thank you, madam. Shall I close the window? Thank you.